For centuries, the ancient traditions of the people of Israel have preserved a unique identity shaped by lineage, faith, and spiritual heritage. Among these traditions, one stands out for its remarkable continuity: the Cohanim, the priestly line believed to descend from Aaron, the brother of Moses and the first high priest according to biblical texts. According to tradition, the Cohanim have passed down their identity from father to son for more than 3,000 years. Their role was not only religious but symbolic, representing a direct connection between the people and the sacred. For generations, this claim was accepted as part of faith, until modern science began to investigate it. With the rise of genetics, researchers asked a compelling question: could there be a biological trace that supports this ancient lineage? The answer began with the Y chromosome, a segment of DNA passed almost unchanged from father to son. Because of this stability, it serves as a powerful tool for tracing paternal ancestry across long spans of time. If the tradition of the Cohanim held true, many men who identify as such today might share similar genetic markers. What scientists discovered was striking. Studies conducted across diverse Jewish communities revealed a recurring genetic pattern among many men who identify as Cohanim. This pattern, often referred to as the Cohen modal haplotype, suggests that a significant number of these individuals may descend from a common male ancestor who lived thousands of years ago. The implications were profound. For the first time, a long-standing religious tradition appeared to align, at least in part, with measurable scientific data. But as with all scientific findings, the reality is more nuanced. Not all modern Cohanim share this genetic signature, and some individuals outside the priestly designation also carry similar markers. This indicates that while there is a statistical pattern, it is not definitive proof. Over centuries, factors such as conversion, migration, and intermarriage have shaped and diversified the genetic landscape. Moreover, genetics cannot assign names to ancient DNA. It cannot confirm that the shared ancestor was specifically Aaron. What it can do is point to the possibility of a shared lineage within a certain historical timeframe. This intersection between evidence and limitation creates a fascinating tension. On one hand, science offers data that seems to support an ancient narrative. On the other, it leaves critical questions unanswered. Are we witnessing indirect validation of biblical accounts? Or are we observing genetic continuity shaped by geography and shared origins? Beyond the science, the story of the Cohanim speaks to something deeply human: the desire to connect with the past. The idea that a lineage could persist across millennia, surviving wars, exile, and cultural change, is extraordinary in itself. Today, advances in genetic research continue to deepen our understanding of human origins. New technologies allow scientists to analyze DNA with greater precision, reconstructing histories that were once beyond reach. Yet even with these tools, there are boundaries that science cannot cross. Faith, tradition, and identity cannot be reduced to genetic sequences alone. The mystery of the Cohanim lies not only in their DNA, but in what they represent: a living bridge between past and present, between the tangible and the spiritual. It is a reminder that some truths exist not just in data, but in meaning. And in that space, where science meets tradition, the ancient priestly lineage continues to endure, not only in blood, but in history. Throughout history, few figures have had an impact as profound as Jesus Christ. His life, teachings, and spiritual legacy have shaped billions of lives for centuries. Yet in the modern era, a new and unexpected question has emerged: is it possible that any genetic evidence of Jesus exists today? At first glance, the idea may seem surprising, even controversial. Genetics is a science grounded in physical evidence: DNA samples, biological remains, and verifiable sequences. In contrast, The story of Jesus has been preserved primarily through ancient texts such as the Gospels, where the focus is spiritual rather than biological. This creates a natural tension between two very different ways of understanding reality. One of the greatest challenges in this discussion is the absence of confirmed physical remains. Unlike many historical figures, there is no widely accepted scientific evidence of relics that can be definitively attributed to Jesus. Over time, several objects have been venerated as sacred, but their authenticity remains heavily debated. Among the most famous is the Shroud of Turin, a linen cloth believed by some to bear the image of a crucified man. Scientific studies have attempted to analyze possible biological traces found on the fabric. However, results have been inconclusive and often disputed due to contamination, the age of the material, and limitations in testing methods. Another key element in this discussion is the nature of Jesus' birth itself. According to Christian tradition, his conception was miraculous, introducing a unique theological dimension. From a genetic standpoint, this raises complex questions. How would such an event be interpreted biologically? Could it even be analyzed using current scientific tools? Some researchers have proposed hypothetical scenarios. If a verified biological sample, such as blood, tissue, or any genetic material, were ever discovered, it could potentially be analyzed to determine geographic origins, physical traits, or even possible lineage. Yet another major obstacle remains. There is no reliable historical evidence that Jesus had children, which limits any attempt to trace direct descendants. Beyond the physical evidence, the fascination with this topic reveals something deeper. It is not only about finding DNA, but about understanding the connection between the divine and the human. For many believers, Jesus represents the meeting point of both realms. Attempting to analyze him through genetics can be seen as an effort to translate the spiritual into the language of science. However, this approach carries risks. Science has clear boundaries. It studies what can be observed, measured, and repeated. Faith, on the other hand, operates in a different domain, where meaning is not always dependent on physical proof. Confusing these realms can lead to misleading conclusions or unrealistic expectations. In recent years, advances in genetic technology have allowed scientists to reconstruct DNA from ancient human remains, sometimes thousands of years old. These breakthroughs have revolutionized archaeology and anthropology. Yet even with these tools, the case of Jesus remains exceptional, primarily due to the lack of verifiable material and the singular nature of his story. So, does the DNA of Jesus exist? From a strictly scientific perspective, there is no confirmed evidence. From a faith perspective, the question may not carry the same importance. And between these two worlds lies a space filled with mystery, where the questions themselves may be more powerful than the answers. Perhaps the true value of this search is not in discovering a genetic sequence, but in exploring the limits of human understanding. In that space, where science reaches its boundaries and faith begins, emerges one of the most fascinating questions of our time. How far can we go in our attempt to understand what transcends the material world?